Hello everyone, welcome back to Microbiome Solutions. Today we are going to see about vaccines. Vaccines are important and we need to understand the immune mechanism that delivers protection against different organisms. This understanding guides us the design of more effective vaccines which is in use. So the best way to confer immune resistance against a pathogen is to mimic the pathogens without causing disease or to devise formulations which mimics its characteristics. Basically, when a microbe is initiating an infection in a human body, the same infection is initiated in order to confer the immune resistance by an alternative such as vaccine. The history of immunization or vaccination, it was first recorded an attempt at immunization which occurred in the 6th century in China. Drying and grinding up smallpox scabs and blowing them with a straw into the nostrils of immunocompromised family members. By the 10th century, this practice had changed to a deliberate inoculation of dried pus from smallpox pustules of one patient into the arm of a healthy patient. So, this will in turn protect the healthy person from getting the infection by smallpox. This method was used in part of the far east of centuries before Lady Mary Montagu brought it into England in 1721 from Istanbul. This is about the history of smallpox, and eventually first really effective vaccine was developed by the English physician Edward Jenner in 1796. He is known to called as the father of immunology or father of vaccination. Now, while many advances have occurred in the last 2 centuries, science is poised for even more in the future. Today, the researchers are currently working to improve existing vaccines and to search for vaccines that fight against HIV, AIDS, cancer and other deadly diseases. The term vaccine is actually derived from vacca, which is meaning cow, since Edward Jenner used a cowpox virus, that is vaccinia, to prevent smallpox infection. So the scientists had used cowpox virus to prevent smallpox infection. The vaccine is an immunobiological substance designed to produce specific protection against a given disease. So this is a biological compound which has got an immunological property which will protect against a disease. And the vaccine is antigenic but not pathogenic. Though it is obtained from the organism, it has got the immunological properties or not the pathogenic property or the virulent property. The virulent property is attenuated or it is disabled in a vaccine. Vaccines have profoundly reduced the prevalence and impact of many infectious diseases that were once common and often deadly. The route of administration commonly used for the vaccines are deep subcutaneous or intramuscular route for most of the vaccines, oral route for Sabins, that is polio vaccine, and oral BCG vaccine, intradermal route again for BCG, scarification for smallpox vaccine, intranasal for the live attenuated influenza vaccine. In the scheme of immunization, there are two schemes currently available for the vaccination, that is primary vaccination and the booster vaccination. So during the primary vaccination, one dose of vaccine, for example BCG, variola, measles, mumps, rubella, yellow fever, and multiple doses of vaccines like polio, diphtheria, pertussis, tetanus and hepatitis B. These are the primary vaccination for one dose vaccination as well as for the multiple dose vaccination, and booster vaccination is given to maintain the immunity level after it declines after some time has elapsed. Example for DT and MMR. So we usually take DT or TT once the time period is over. That is called as a booster vaccination. Overview of immune response. When a microbe enters the body, the immune system responds in an attempt to eliminate the infectious organ or infectious agent. So the organism is entering inside the body, and our human body or the host body detect it as a foreign particle and try to fight or try to eliminate the infectious agent from the body. For that, innate immune system relies on the immediate recognition of the antigenic structure, which are common to many microorganisms like pathogen assisted molecular pattern or PAMS, an adaptive immune response made up of T cells and B cells that have a unique receptors to the specific microbial antigens and take time to respond. So innate immune system is nothing but the immune system or the immunological cells that one person has derived by the birth. And adaptive immune is nothing but the acquired immunity that a person is getting during his or her lifetime. So innate immunity is spontaneous or which will act faster when the pathogen enters the body, whereas the adaptive immunity which will bind to the or which will respond to specific organism or specific cells, it will produce only specific B or T lymphocytes which has unique receptors, and it takes time to respond to the particular infectious agent. And what is the goal of vaccination? Basically, the goal of vaccination is to generate and sustain the number of antigen specific B and T cell against a particular pathogen so that the antibodies are produced. Antigen sufficient to provide the protection against that particular infection, and most of the successful vaccines are against small organisms, that are viruses as well as the bacteria, and microorganisms have evolved complex defense mechanisms that interfere with the immune response. Some of them are molecular mimicry, interference with the antigen processing, prevention of apoptosis of infected cells. Properties of an ideal vaccine. How to choose or how to say that a vaccine is ideal to be used. So it should give lifelong immunity and broadly protective against all variants of the particular organism and prevent the transmission of disease from one person to the other, and rapidly induces immunity and effective in all subjects irrespective of the gender or irrespective of the status of the patient, whether it is old or very young, whether it is male or female, it should be effective in all the subjects. And it should transmit maternal protection to the fetus. You can see in this picture the benefit of vaccination. See, from the first lamb who is protected against the first picture, the first lamb is protected against with the vaccination, and the second, the child lamb is taking colostrum from the mother who is protected against the infection, and the final lamb is surviving, whereas the second picture is showing the consequences of non-vaccination when the baby is or when the lamb is taking the colostrum without having any protective or without having any immunological cells, and the baby is falling sick or the baby lamb is dying because of immunocompromised status. So it should require few immunization to induce the protection, not many immunization schedules. No need to be administered by injection. It can be or it should be oral, intranasal or transcutaneous, and it should be stable, cheap and safe. Development of immunity in infants. So infants' immune system is relatively complete at birth. That IgG antibodies received from mother are important for the protection of the infant while the infant is developing its own repertoire of antibodies. See, basically when the child is born, the child is getting IgG antibodies from the mother through the placenta and from the breast milk IgA is received. So the passive transient protection by IgA against many common illnesses is also provided to the infant in breast milk. So through the placenta IgG is given from the mother to fetus, through breast milk IgA, that secretory IgA is given to the baby. What happens in a natural infection to produce immunity? That is natural active or natural passive immunity. So it is to develop a vaccine we must first consider that what happens in a natural infection to produce protective immunity. So this is called as a correlates of protection. That what is the benefit of causing a natural infection which in turn produces lifelong immunity to the patient, as like happens in chickenpox. So an effective vaccine against intracellular pathogens should only induce effector mechanism, ultimately leading to the destruction of the parasites or the disease causing agent, and the vaccine should not trigger components of the immune response favoring the survival of the microbe. So there are different types of vaccines available. First one is a killed microorganism vaccines. These are previously virulent microorganisms that have been killed with chemicals or heat. So the virulence property is not seen in this type of vaccines. Next is live attenuated microorganisms. So here the microorganisms are live but the virulent properties are disabled. So they typically provoke more durable immunological responses as the organism is alive, which are the preferred type of healthy adults. Next is the toxoid. They are inactivated toxic compounds from microorganisms in cases where these toxins rather than the microorganism itself causing illness. Next is subunit vaccine. A fragment of the microorganism can create an immune response, where example is the subunit vaccine against hepatitis B virus, that is composed of only the surface protein of the virus which are produced by in the yeast. First one is the live vaccine. Live vaccines are made from the live infectious agents without any amendment or any changes in the organism. The only live vaccine is variola, that is the smallpox vaccine made from live vaccinia or the cowpox virus. We have seen that initially Edward Jenner used cowpox virus to treat or to produce vaccine against smallpox, which is not pathogenic but antigenic, giving cross immunity for variola. So the vaccine is made of vaccinia virus, which is giving protection against variola virus. Next is live attenuated vaccines. Here virulent pathogenic organisms are treated to become attenuated and avirulent, but they are antigenic. They have lost their capacity to induce disease, but they can retain their immunogenicity. So the virulence property is disabled and immunogenicity is activated. So these vaccines may be given by injection or it can be given via oral route. And administration is orally, intradermal, intranasal or subcutaneous. Examples include vaccines against Vibrio, Salmonella, BCG, polio, Japanese encephalitis, yellow fever, influenza, mumps, measles, rubella, chickenpox and smallpox. Advantages of these includes it stimulates the generation of cellular as well as humoral immune responses. Since these can multiply inside the host, single administration of vaccines produce long-lived immunity. So this organism which has disabled virulence property with immunogenicity is multiplying inside the host without causing any infection. So single administration can provide long immunity. Multiple booster doses may not be required and oral preparations are less expensive than giving injections. Elimination of wild type virus from the community. Disadvantages are may very rarely convert to its virulence form, causing diseases, and live vaccines cannot be given to immunocompromised individuals, can cause serious illness or death in the vaccine recipient if the patient is immunocompromised. Since they are live and because their activity depends on their viability, proper storage is critical. So the cold chain has to be maintained when the vaccines are transported. If it is not maintained, the vaccine may not be viable. Next is the killed vaccine. When it is unsafe to use live microorganisms to prepare vaccine, they are killed or completely inactivated. So this organism are killed or inactivated by the treatment with using heat, formaldehyde or gamma irradiation so that they cannot replicate at all, but it remain antigenic. So inside the body it cannot replicate, but it can have the antigenic property. Usually it is safe but less effective than live attenuated vaccine. Live attenuated vaccine can multiply or replicate periodically to provide lifelong immunity. Routes of administration of killed vaccines are generally intramuscular or subcutaneous, and examples include against Salmonella typhi, Vibrio cholerae, Yersinia pestis, Bordetella pertussis, poliomyelitis, Salk vaccine, Japanese encephalitis virus, rabies virus, influenza virus and hepatitis A virus. Advantages includes it is safe to use and can be given to immunodeficient and pregnant individual, cheaper than live attenuated vaccine, and storage not as critical as live vaccine. Disadvantages includes microorganisms cannot multiply, so periodic boosters must be given to maintain the immunity as it is a killed vaccine. Only humoral immunity can be induced, and most killed vaccines have to be injected, and some vaccines such as Bordetella pertussis induce ill effects like post-vaccinal encephalomyelitis, but it is very rare. Inactivation such as formaldehyde in the case of Salk vaccine, which is polio vaccine, may alter the antigenicity of the virus. Next is the subunit vaccine. Subunit vaccines contain purified antigens instead of the whole organism. So such a preparation consists of only those antigens which can elicit protective immunity. And subunit vaccines are composed of toxoids, subcellular fragments or surface antigen, and the effectiveness of subunit vaccines is increased by giving along with the adjuvant. Adjuvants are nothing but the chemicals which are used to increase the immunogenicity of the vaccine. The route of administration remains intramuscular, and the examples of subunit vaccine include cell wall polysaccharide of Haemophilus influenzae, Neisseria meningitidis, Streptococcus pneumoniae, Group B Streptococcus, Salmonella typhi, and toxoids like diphtheria and tetanus, membrane proteins of influenza virus and hepatitis B virus. Advantages are it can be safely given to immunocompromised people and less likely to induce any side effects. Disadvantages are since it is subunit, antigens may not retain their native conformation, so that antibodies produced against the subunit may not be recognized by the same protein on the pathogen surface. So isolated protein does not stimulate the immune system as well as the whole organism vaccine, because the whole organism will stimulate the entire immune system, where this subunit, a part of the organism, will have limited immune activity in the host. Next is the conjugate vaccine. It is a type of vaccine that is created by joining an antigen to a protein molecule, and usually it is used to immunize babies and children against bacteria which has got a polysaccharide capsular membrane. And the immature immune system of the baby or very young children often have difficulty in recognizing because their immune system is not well established. So ordinary vaccines may not be effective for some diseases. Thus conjugate vaccines are used by joining antigen along with a protein molecule. A conjugate vaccine therefore usually consists of a polysaccharide antigen which is combined with a carrier protein, and the combination of this antigen with the protein creates a substance that is more easily recognizable to the white blood cells in the blood, resulting in the stronger immune response. Because the infant's immune system is not well established, it is used as in combination. Examples include tetramune vaccine, which combines tetanus and diphtheria toxoids, whole cell pertussis vaccine, and H influenza, Haemophilus influenzae type B conjugate vaccine. And next is the recombinant vaccine. These vaccines are produced by using recombinant DNA technology or genetic engineering. Recombinant vaccines are those in which the genes for the desired antigens of a microbe are inserted to a vector. For example, hepatitis B, diphtheria, cholera and tetanus. Advantages include these vectors that are not only safe but also easy to grow and it can be stored. Antigens which may cause damaging responses can be eliminated from the vaccine. Examples include cholera toxin A, which can be safely removed from the cholera toxin. Cholera toxin has two subunits A and B, among which A subunit can be safely removed from this CT. Disadvantages: since the genes for the desired antigens must be located, cloned and expressed efficiently in the new vector, the cost of production is high. When engineered, vaccinia virus is used to vaccinate, but care must be taken to spare immunodeficient individual. And next is the DNA vaccine. DNA vaccines are being hailed as the most compromising or most promising of all over the newer approaches to immunization. These vaccines are still in the experimental stage. Like recombinant vaccines, genes for the desired antigens are located and cloned. DNA vaccines have to be induced both humoral and cellular immunity, which in turn will produce stronger immune response against the particular infection. Route of administration by gene gun method, intradermal and jet injection. Examples: bird flu DNA vaccine. Most of the DNA vaccines are in clinical trial phase. Advantages: DNA is very stable, hence storage and transport are easy. DNA sequence can be changed easily in the laboratory, and inserted DNA does not replicate and encodes only in the proteins of interest will do so. Disadvantages: induction of autoimmune responses like anti-DNA antibodies may be produced against the introduced DNA, and induction of immunologic tolerance. The expression of the antigen in the host may lead to specific non-responsiveness to that particular antigen which is inserted. And next is the anti-idiotypic vaccine. This is a unique amino acid structure in the antibody that is called as an idiotype, which can be considered as a mirror of the epitope of the antigens, or which act similar to the antigenic epitope. So antibodies can be raised against this idiotype by injecting the antibody into the another animal. So this anti-idiotype antibody from the other animal mimics part of this three-dimensional structure of the antigenic epitope, and this can be used as a vaccine. So when the anti-idiotype antibody is injected into a vaccine, the antibodies that are basically anti-idiotypic antibodies which are formed and recognized as a structure that is similar to a part of the virus and might potentially neutralize the virus. Advantages: these antibodies protects against potentially significant antigens can be produced, and disadvantages are only humoral immunity is produced. There is no cellular immunity and has poor memory. So each time you will have to give the vaccine to give lifelong immunity. Identification and preparation of idiotypes is labor intensive and it is often difficult. And the vaccination for travelers, it varies according to the country of arrival and departure. So common vaccines according to the country travel to are TAP, yellow fever, cholera, meningococcal, pneumococcal, HIV, influenza, rabies, plague, Japanese encephalitis, tick-borne encephalitis, measles, hepatitis B, hepatitis A, tetanus, poliomyelitis, typhoid. And Hutch, for instance, necessitates meningococcal vaccination from all over the world, and yellow fever from places like South Africa and cholera from places like India. So these are the must for travelers. Stages of immune response. When we look at these stages: initiation of immune response, development of immunological memory, deciding on appropriate immune response for protective vaccines. This is a current challenge, is to achieve strong immunogenicity without increasing the incidence of adverse effect to the vaccines. And initiation of immune response, an antigen must be recognized as foreign, that is a danger signal, and then initial recognition is by dendritic cells and tissue resident macrophages in a non-lymphoid tissue, and it is uptaken by the antigen processing, migration of cells to the lymph node and maturation of the dendritic cells. Thus the immunological response is activated. So initiation of immune response, that is antigen processing. When the antigen entered cells by endocytosis, such as the bacterial cell, which are broken down in lysosomal vesicles, and these antigenic peptides are loaded into the class 2 MHC molecules so that it is taken to the cell surface of interest. So antigenic peptides along with the class 2 molecules, class 2 MHC molecules, are taken to the cell surface, where these antigens synthesized in the cells such as viruses are broken down into peptides by these proteosomes and transported to rough endoplasmic reticulum for loading into class 1 MHC molecule and transported to the cell circle. So class 1 molecule and class 2 molecules are transporting different types of cells. So thus surface expression of MHC molecule increases. And next is the migration and maturation of the cells. Antigen presenting of the dendritic cells migrates from the tissues to the draining lymph nodes, and migration is controlled by chemokines and the chemokine receptors. Next, dendritic cells mature to display more of the surface molecules that are needed for the interaction with the T cells, like CD40, B7 deliver co-stimulatory signals to activate the T cell. And development of immunological memory. Almost all vaccines have the objective of long-lasting protective immunity. Not certain how to achieve this, but it is proved. Memory populations of cells have encountered antigen and changed phenotypes as a result of the stimulation, and phenotypically they are defined as in-memory cells that are shown to divide more rapidly than the naive cells. So there are constraints on the duration of the development of immunological memory. And hazards of immunization. No immune response is entirely free from the risk of adverse reactions or remote sequelae. The adverse reactions that may occur may be grouped under the following heads: reactions inherent to inoculation, reactions due to faulty techniques, reactions due to hypersensitivity or neurological development, or the provocative reactions. These are the different hazards that can cause due to immunization. So this is about today's lecture. If you have any doubts, please put it in the comment section. Please do like, share and subscribe to our channel. Thank you.